Hey guys, today we're going to be talking about the sulfur indole motility medium, or what we simply just call the SIM medium. The SIM medium is a combinational differential medium used to differentiate bacteria based on their motility, their production of indole, and their production of hydrogen sulfide gas. So kind of with that in mind, let's talk about the setup of our medium and kind of how each of these different components to it work. So the first thing we'll talk about is the motility. So our medium is a semi-solid medium, so you can kind of see here again it doesn't fall down when I turn the tube over. So it is made with agar, specifically generally around 0.35% agar concentrations. And that 0.35 agar concentration is high enough to slow down the spread of non-motile bacteria, so you would only see growth along the stab line, whereas motile bacteria will grow all throughout the tube, making the entire tube cloudy. The next thing we then tend to look at is sulfur reduction. So the medium has ferrous ammonium sulfate and sodium thiosulfate in it. And any bacteria that has enzymes that can break down either one of those products will produce hydrogen sulfide gas. Now, the hydrogen sulfide gas is then going to bind to the ion components of the ferrous ammonium sulfate, producing ferrous sulfide. And ferrous sulfide becomes a black precipitate. So any bacteria that can reduce sulfur will turn the medium black. Our last thing is then the indole production. So the medium also has the amino acid tryptophan. And if a bacterium has the enzyme tryptophanase, it can break tryptophan down into pyruvic acid, ammonia, and indole. So the way that we actually test to see if indole is present is that we add Kovacs reagents to our tube after incubation. And this is really important. If you add the Kovacs reagent right after you inoculated it and then put it and set it away to incubate for 24, 48 hours, you ruin the experiment. You have to wait until you come back after the incubation period, or 24 to 48 hours, to add the Kovacs. But the Kovacs come in a bottle kind of like this, where you can just kind of press open the top and you can add it directly to the tube. Or it might come in a bottle kind of like that, where you have to use a pipette to add it to the top of your solid media here. And you're looking for normally about 5 to 10 drops will give you a layer kind of about that size. Not a whole lot in there, but just enough to get a nice layer of the Kovacs on top. So with our media basics kind of out of the way, let's go ahead and talk, results. So after you've inoculated your sample, you've waited your 48 hours or so, or a week or so, depending on how long we're incubating, and you're coming back to look at the results and scoring your tubes. The very first thing to look for is going to be hydrogen sulfide gas production. Now, of course, when I grew up my samples, I couldn't get one to turn black, but just real fast, here's a picture of what that looks like. And let's go ahead and get that out of the way. So if your tube turns black, that's going to be positive for hydrogen sulfide production. If it stays our agar color, then it's going to be negative for hydrogen sulfide production. The next thing that we would then be looking at is going to be motility. So when you come back and you look at your tubes after you've incubated them, if you have growth just along the stab line, kind of like you can see on this tube here, that would be negative for motility. But if it looks kind of cloudy all the way through, kind of like in this tube here, that would be positive for motility. Kind of on that note, if your tube turns completely black because of the hydrogen sulfide production and you can't actually tell whether it's motile or non-motile, let me know and I'll tell you what result you're supposed to be getting. Last thing to talk about then is again the indole production. So after you add the Kovacs, again make sure to add it after the incubation period, not before the incubation period. If your tube comes out looking kind of clearish or yellowish like that, that's going to be negative for indole production. But if it turns this nice red color, then that's going to be a positive result for indole production. In terms of inoculating the tubes themselves, you have to use a stab technique. So again, we have to use our stab technique in order to inoculate this medium, and you want to make sure that you use a needle, not a loop, to do the stab technique with. It's generally best to do the stab technique from a broth, so a broth to our deep here, but you can also do it from a slant or from a plate if you have your sample on a slant or a plate. It just tends to look nicer and come out better if you do it from a broth. But with that in mind, that should be everything that we need to know for our sulfur indole motility meeting.